Welcome. I'm Kristy Deiner, and it's an absolute honor and pleasure to be with you today. And I was tasked with talking about the challenges that we've overcome in the last 4 years and the opportunities we have for using environmental DNA in biomonitoring of aquatic resources. But before I get to that, I just want to acknowledge the efforts and the dream that probably was in these two fine scientists' head quite some years ago to bring us all together and form the DNA Aquanet. And I don't know the true origin story of how this all came about, but if this is how it started, this is how it's going. There are over 400 members in 39 countries, and potentially over 1,000 of you registered for this conference and are listening. And it is a truly inspiring time to be doing what we're doing. And it is with the effort and time and responsibility that we all feel for, advancing the technology to really understand what's going on with life on our planet. And I just want to say thank you to all of you for listening and for also participating over the last 4 years. So as we jump in, I'm going to address the elephant in the room, which is, what is environmental DNA? And as you may know, there's quite a few of us having a discussion in the literature right now. And we're really trying to figure out, sort of how to deal with the complexity of what environmental DNA is. In the broadest sense, this is just DNA that's been sampled from the environment in the form of a sediment sample, a filtered water sample, or even a plankton net. And this gray area here in terms of the size of what gets found in these environmental samples means that sometimes we're actually sampling DNA that's inside an organism, this organismal DNA component. But there's also this extra-organismal DNA component made up of gametes, cells, organelles, particle-bound DNA, and free DNA. And it's this portion that's really interesting. One, because it's actually separated from its organism and potentially separated in space and time. Therefore, the inference that we can make when we're looking at this part of environmental DNA is different than when we actually have the whole organism sampled in our environmental sample. Meaning that we have to be conscious that there's an uncertainty associated with this portion. So today when I'm talking, most of what I'm going to be talking about is how we rein in the uncertainty and do a better job at understanding extraorganismal DNA in the environment and how we can use this for biomonitoring. So I think for all of us, it is clear the potential of using environmental DNA to do monitoring of species and processes and, sort of agricultural and aquacultural systems and all their impacts and all the other aspects that we need to understand about how our sort of ecosystems are functioning and how we restore them. And I really highly encourage you to check out this new review by Barry et al. They really did an excellent job of summarizing this and it sort of gives us the capstone of where we are. And now for the rest of my talk, I'm going to jump into sort of the nitty-gritty details of how we do this from water. So I'm going to focus on kind of 3 drops of the whole sort of field of eDNA biomonitoring from water, and it's really been the focus of the DNA Aquanet over the last 4 years. So I'm going to talk about sampling and standards for workflow and reporting and how we create really reliable detection methods. And here I put bio in parentheses because from a traditional sense, biomonitoring is really about using a species or a community to infer a state for an ecosystem. And more broadly, we are just talking about biodiversity monitoring as well. We have made great strides in understanding how to sample different ecosystems. And when it comes to rivers, what's been really exciting is just in the last year or so, there's been these incredible detailed work about putting caged individuals in a river and then looking at how that DNA from that source is dispersing depending on how the river flows and in seasons and all kinds of different aspects, and sort of really looking at the density of where we find the DNA detections. Now, these kinds of detailed studies are really important for us to understand how that extraorganismal DNA is behaving in the environment. But, the conclusion from a lot of this is just to enable comparisons over time in rivers, we need to incorporate these hydrological conditions, but also these species traits, so the shedding of DNA and how it's getting in and what state it's actually in, and I'll talk about that more later. And when we move into lentic systems like lakes and ponds, there's been some great studies that have really gone in and just oversampled the systems and sampled them in ways that we can combine them and try to understand the spatial impact of how we sample these systems. And here, you know, this was a great study in the UK looking at this lake, and they looked at different depths and the shore and where all these samples came from. This one also really sampled the surface waters, and what you can see is you get different numbers of species detected depending on sort of where you take these samples. And the take home from this is at least sample the perimeter, right? But there's lots of other things that come into play here. These systems are cycling, there's stratification, there's other considerations, not just in space, but also time. So now I want to talk about standards for workflow and reporting. And I have a couple Sankey diagrams up here just to show us, you know, it's kind of, I don't know, the Wild West out there, right? So when you do a review of the literature, you see that depending on, you know, the taxa and the collection and extraction, and, you know, we're kind of all using different methods out there. And this creates concern because it's, you know, how does this, you know, all play out? What's the role of all of these decisions and changes and protocols and all of this, right? And I've tried to break this down into what I think are the three main choking points. You know, we can go out and we capture the DNA from in our environmental sample, and that really results in the DNA that's sampled. And then there's this sort of method that we use to purify and isolate that DNA from that water. And we end up with purified DNA. And then we choose a detection method. So either we're looking for a specific species or we're looking for a whole community. And that process also plays into the detection rate, right? But all together, these things are not independent and they lead into each other. And so I think it's really important that we focus on these choking points, the sampled DNA, the purified DNA, and the targeted DNA, and try to optimize our methods in this direction. And that's because, you know, sort of the myriad of studies that have come out that show us that method choices are interdependent. You know, I sort of call this the effects of literature, right? Which shows us that if we change any little part in this process, we actually can have an effect on detection rates. So for example, this study we did in the marine system, it was using, you know, a community eDNA metabarcoding approach, and we looked at different filter types, we looked at different extraction methods. The color here represents the different extractions, and here's the detection proportion in our replicates. And this is an invasive species, and just by changing our methods, you know, sort of, you know, extraction methods, we could increase going from down around, you know, 10% detection proportion up to almost 80%. That's a big difference. And so we need to understand which of these sort of processes really leads to those big differences. Now, there's going to be small changes as well, and maybe those are not so important to focus on. But what it really means is that there's going to be different methods used, and different laboratories are going to use different ones. And, you know, this publication was the result of a bunch of us in the DNA Aquanet getting together and trying to think about how do we synergize all the methods that are being used for macroinvertebrates, and can we come up with these kind of calibration experiments where we compare methods using the same DNA across laboratories with a standard protocol versus a protocol that might be your pet protocol that you use in your laboratory, and really compare and figure out where the variation is. And unfortunately, due to COVID, we haven't been able to execute this experiment, but it's going to get underway, you know, and so we'll make advances like this. And we really want to encourage everyone to start thinking on this scale. If we're going to generate monitoring tools using DNA-based methods, we need to really have these calibration experiments. So now I want to shift to talk about, you know, what really makes this a reliable method. And that all relies on how much detections are consistent in space and time, and there's some really great studies coming out that are looking at these elements and kind of categorizing, what happens when you sample the same location over many weeks, and what does the technical replication do at a single time point, and what information you get from that. And it's great to see that, maybe some things are not as important as other things in terms of figuring out what's similar and not similar. And so to capture diversity in space and time, we need to think about these elements. The other major achievement, and I just really want to say hats off to combining both a calibration study and really trying to give us what we need in terms of setting up a reliable standard for how we do reporting. And this goes into qPCR and understanding limits of detection and what we report, and why in eDNA we might do something a little bit different than analytical chemistry, because sometimes we're dealing with an endangered species that this might be the last potential population. And so while this data is not trustworthy from eDNA, we still might want to report that because it could tell us something about the species and the environment, and we should throw all the other methods we have if this really is sort of a conservation concern. And to build on all of that, there was a large group of us also that got together in a workshop and tried to really assess, how is it that we can say an assay that's been developed for eDNA, and this is again qPCR and single species detection, is operational. How can we take that assay and start applying it for management? Now, not all assays are created equal, and some are validated to very different levels and aspects. And so one thing that is absolutely clear is that when you want to use something for environmental DNA, the assay is the sum of all the parts. You can't just take, and many of us probably learn through experience, just the primers and the probe out of the literature and then just go start applying it, because all of a sudden you get a bunch of things detected that aren't the target species, and what's going on there. And well, maybe the original author didn't, was working in a different geographic zone and that species wasn't co-occurring. And so, there can be lots of reasons why this doesn't work. And Bettina's going to give a great talk later this week about the in-depth process that went behind developing this, and I really encourage you to go to her talk. So building on this, let's talk about standardization for a minute. And, I want to talk about the complexity of the decisions that need to go into this. So I'm going to do this by walking through an example. So, when we think about the complex state of DNA, it can be in all these different forms. And because we're still uncertain about which form persists in the environment and when and how long, we can maximize detection from all of these, but they're actually sampled in different ways, right? So some can be captured through filtering and some through precipitation. And, after you get that, though, how do you purify that? And so if you have, for example, a particle bound state, you have to do a pretty specific kind of extraction to make sure that's desorbed from that particles, versus the types of lysis reactions you might have to do depending on the cell type and all kinds of things. And, the other major unknown here is the temporal and spatial inference. So with free DNA, we're pretty close to understanding this. We see that it's pretty fast, and it also depends on bacterial demand. So they're eating DNA, but particle-bound DNA is potentially very stable. And the current state of what's going on with organelles and cells, I would say we have a lot more research to do. And to top that, water chemistry matters, right? And so many of you who've gone out, you go out in different water types from clear, to murky, and, there's all these other inhibitors that can get co-extracted along with these, particles that DNA can actually get bound to. And the kicker is, the DNA is not just co-extracted with inhibitors, it's also getting coming into more contact, because when you filter things, you basically are filtering out those sediments and creating an environment where DNA can bound to that. So now imagine lysing your cell and having a whole bunch of sediments on your filter. What do you think is going to happen? It's really important to understand how all this chemistry is working. And to that extent, I think one of the most important things is that and you may not want to hear it, we need ecosystem-specific parameterization of eDNA. And I put methods in black here because that's my contribution. And if all of you read Harrison et al., you maybe cringed at this because you're like, well, how are we ever going to get to standardization? But the reality is we're detecting a molecule and it has chemistry. And so, getting standard detection rates has to take into account the chemistry. And if that isn't enough and your head isn't spinning, I want to announce that it's been really fun to do this project. So this is sort of the capstone project over the last 4 years that Working Group 3, which was, talking about field and laboratory methods, and there's 27 of us that arm wrestled for 4 years to really sort of bring this into practical terms and try to make sense of all of this. And this is culminated in 98 pages chock full of how to make all this practical. How do you make these decisions when, there's a cost or a time or, sort of a people problem? There are all these trade-offs, and they are real when you come to, and they're very real when they come to designing, monitoring systems. So stay tuned. This is so big, it's going to be an ebook published soon. And Kat is going to dive into this later in the week and talk a little bit more about it. So, this resource is going to also hopefully be something that can be updated in time and as we learn more, and we're really looking forward to the response of the community as well as growing and contributing to it in the future. So for the last minute or so, I just want to talk about, the fact that eDNA, the science that we're doing, is done in interdisciplinary teams. And this is just a quick literature search of author affiliations and sort of what broad categories you can put them in. And, it sort of gives you an idea of who your dream team should be, and I really think bringing manager, a policymaker, ecologist, a molecular biologist, environmental biochemist, hydrologist, and data scientists, that's what we need to bring together. And we'll be able to design the monitoring programs of the future using this technology. So what's next? Well, if you missed this review, it's an absolute one you should check out. And it really goes into sort of the populational and functional inferences that we can potentially make from eDNA and eRNA. So when we wanted to get into function, this is a whole new area, and it's a really exciting time to be thinking about how we can move forward, not only advancing sort of the nitty-gritty details of application, but that there's even new technology that's going to come on board and allow us to answer all sorts of questions that we can't do just with mitochondrial DNA. The next challenge we have in the future is how do we scale all this up? And so I'm really excited that I'm going to be leading a global sampling campaign for sort of testing this crazy idea I have that, there's these places on Earth that are accumulating environmental DNA, and that we can actually predict and seasonally sample these to get essential biodiversity variables. And so while the local scale is incredibly important for restoration projects and really trying to change the path that we have in front of us, we need to know at a global scale that that's all working synergistically to put us on a different trajectory. And so the value of how we can do this with the same tools is really exciting. And so you'll hear a lot more from me about participating in this global study. And last but not least, I'm really excited to launch with you today the beginning of simplexdna. And there's so many companies starting and getting out there and offering these services, and we need more and more of them every day, because we cannot let this technology stay in the ivory tower. It's got to get out there. And as I said in the beginning, we're at a time where we're working on a technology that can be implemented simultaneously, and we need to build up this infrastructure that has the capability to execute this. So once we get our policymakers and managers on board, we need to be able to offer this, and there's really so much room out there for growing this, and we're excited to jump in and offer what we have as a team. And our goal is just to really build DNA-based monitoring solutions to save life on Earth. And if you're interested in this endeavor and you want to talk more, I'm really happy and excited to do that with you. And with that, I would just like to say thank you for your virtual attention. And if there's time, I'll take questions in person now. And if you want to reach out to me, you can find me on Twitter at alpineedna or on Gmail. So thank you so much for listening.